In this second session, I am checking for whether I can still follow up on the first session that I did. If it's still equally enjoyable and productive for me, whether it is a worthwhile pursuit like it was yesterday. And lastly, I am checking to see if I can still deliver, much like the previous session, um, a good presentation. The key idea here is if I'm natural enough, if I'm spontaneous enough, for my audience to get something out of this session, study session, or whatever this is. And hopefully they will stick around and hopefully they will come back to see more of my stuff. And with that being said, today we are going to cover lecture number 13, Advanced Neurology and Endocrinology. I was going to initially thinking of picking the lecturers strain from lecture 10 to I think lecture 12, which were delivered by I think yes this one delivered by not Sapolsky, and I got his name wrong in the previous video. Apologies for that as well. Sapolsky did not deliver these set of lectures because they were like introduction to neuroscience, like part one. Then this one is part two, and I think there's like one other video which I'm thinking I would watch in my leisure time and not content with that here now, because I want to get the best information, the best some sort of feeling like you have a special lecture, and everything Sapolsky or everything Professor Fink does is quite special. So I want to get that kind of vibe for the content, or this particular session. And once I'm done, maybe I can do some background studies and check those three videos out. That is the idea so far. And with that said, we are on.
Stanford University.
What have we got to now? You guys have all the basics in hand now for our next two buckets, neurobiology and endocrinology. What you have by now, I hope, is a sense of especially the neuro as the final common funnel out of which comes all that behavior stuff we've been thinking about. Genes, whether they are changing in a static and then punctuated way or a gradualist way, what they code for, we now know are proteins. And in the realm of behavior, what that's all about, you now know, are enzymes that make neurotransmitters, enzymes that break them apart, receptors for neurotransmitters, the ion.
I'm going to make a point and emphasize on the need for protein, or the importance of protein, not the need for protein. Um, I feel like one of the misleading aspects of thinking about biology and studying this kind of medical related knowledge is whether or not you put enough emphasis on protein. Because for the longest time, and that happened after I took like multiple courses in biology, and I wonder if this is the same issue for other people as well. We were sold low on protein, because I came out, and this might be a me issue, not a instructor issue of courses. Protein kind of solves everything, from how our neurons behave to how diseases work. So this is a key revelation that happened to me while watching the first lecture of Professor Fink. I think it was the physiology series, in which he basically outlined how a medicine works and how our cells communicate with each other, and how different materials move in and out of the cell. So basically, cells would have something called a receptor, which is a form of protein, binding protein to something, and the chemicals would just travel using that pathway. So very early on, even in this lecture, I would like to call that, like to highlight that for especially people who do not yet appreciate what biology is and how it works. Like there is a mechanical process to it. So uh, if you have not known that bit of information, this is a good time to get into the phase of appreciation with respect to how mechanical the entire process is, and it is very learnable. You can learn it, you can like appreciate the sense making that goes into these causes and effects. Okay. More or less. So I'm going to read this. When it comes to behavior step, we've been thinking about the genes, whether they are changing in static, they punctuated way, or gradualist way. But the code for now we know are proteins. So this is the information. And in the realm of behavior, what that's all about, you now know are enzymes that make neurotransmitters. So enzymes make, I'm taking notes, neurotransmitters. The ion channels that allow all the excitability stuff of all of that should be a way of conceptualizing what all that gene stuff that we have had so far funnels into. Likewise with the neurology. Okay, I'm let him speak.
That allow all the excitability stuff. All of that should be a way of conceptualizing what all that gene stuff that we've had so far funnels into. Likewise with the endocrinology, all those ways in which, reproductive status is going to change how the brain works, all of those great evolutionary models, blah, blah, that's now neuroendocrinology. That's now the ability of hormones to affect the nervous system. And what came through, hopefully, over and over for all of you in the lectures over the last week are two themes, lots of different ways by which the nervous system and the endocrine system can change its function over time. And number two, lots of different realms where there will be individual differences. All the ways in which you will have a brain and a bunch of glands that are different than the person sitting next to you. And what you should be able to piece apart readily by now is where some of those individual differences are coming from in the form of genetic influences, where some of them are coming from environmental ones, to begin to take some of those if-then clauses back in the genetics lectures. If it smells like a relative, then cooperate with them, to begin to imagine now how the nervous system, how hormones would actually translate that stuff into this next bucket. So this will be our last bucket. Today we'll be looking at all the ways in which everything you heard in the last week is vastly more complicated. And on Friday what we will do is focus in on the part of the nervous system that is most pertinent to the rest of the course. Should you ever happen by some chance to stumble into medical school somewhere down the line, when they get around to the nervous system there, you are going to hear about the spinal cord endlessly. You're going to hear about the cerebellum. You're going to hear about parts of the cortex. You are going to hear next to nothing about the part we'll be talking to on Friday. And what it's about is emotion and behavior and affect, a part of the brain called the limbic system. In medical school, they will pound into your head over and over and over how the spinal cord moves different motor systems, because that's 99% of what neurology can concern itself with, because it at least has a fighting chance of doing something therapeutic there. This is the part of the brain that is most defining to who we are, personality, temperament, all of that, and it will be central to making sense of all of the stuff coming in the second half of the course. So it's going to be a very different domain of the nervous system.
I think he's
So
talking
what
about
today is
the
about is taking
spinal
everything you
cord.
learned over the last week, discovering at the ways in which it is tragically distortive and not quite right, and comes with all of these but wait a seconds and qualifiers, and all the ways in which this first pass you had in the last week sets you up now for appreciating an enormously more complicated system than you've already seen, one which generates thus vastly more possibilities of communication, of information contained within these neural pathways, one which generates vastly more possibilities of individual differences and of experience leaving its imprint, one that generates vastly more possibilities for things to go really wrong in the realms of abnormal behavior. Okay, so a lot of what we'll be doing is taking what you got in the last week and seeing where, in fact, current research shows that it is much, much more complicated. Okay, first version. One of the rules that runs through all of neurobiology, you start learning the field, and at some point they pull out the name of this guy Dale. Dale, who was the lawgiver. Dale was some neurobiologist early in the 20th century who generated 2 laws that every single child was forced to learn throughout the 1920s and 1930s about how the nervous system worked. Sometime later than that, Dale's 2 laws, and we will start off with one of them. And it makes perfect sense until you see that, in fact, it has no basis in reality. Here was Dale's first law, which was you got your neuron, and you should know by now we are dealing with our schematic neuron. And once we get into circuits in more detail, we will give up the diamond shape on the neuron cell bodies and switch over to circles, showing just how fast evolution can be. But we've got the neuron here. And what we've got are the axon and the axon terminals. And what Dale's law number
Yes, and something that I would like, why did I say yes? Yes to Professor Sapolsky, yes. Okay, one shorthand that I conjured up yesterday while watching, I think, lecture number 10 is how to memorize accent and the shape of it, because like the new not accent but neuron and the shape of it. Okay, so this is basically the hand, and the hand touching something and sensory information basically going. These are the fingers are the dendrites, and this is the axon and axon helix or whatever, and the information is passing through your hands because this like tentacly stuff and going to this, and this is like maybe the terminal axon terminal where the information is sent to other neurons and stuff. So I just felt like pointing that out. It will help people, and I want to help people. Yay.
2, and of course it's irritating
Cringe. I should never yay.
that we're doing number 2 before number 1, but tough. What you've got here with Dale's law number 2 is each neuron has one characteristic type of neurotransmitter. One and only one, and that's what it's releasing from all of its axon terminals. And thus, you could categorize any given neuron as this is one that releases serotonin, this one's one that releases dopamine, et cetera, et cetera. Notice.
Let me get this straight. So each neuron basically deals with or sends out just one sort of neurotransmitter. So for instance, if a neuron gives out serotonin, it is not going to give out dopamine. Release is the word. So Gale's law 2, Gale's second law, something that I have to Google, is each neuron releases one particular neurotransmitter. Question mark. Okay.
World of difference as to what types of neurotransmitters it has receptors for. That's something else entirely. That's what neurons over there it's listening to. But any given neuron is releasing only one kind of neurotransmitter. So that one went down the tube during the 80s with the discovery, in fact, that there are multiple neurotransmitters released by neurons. And this was boggling to people and forced people to give up on Dale number 2. And initially this just seems like pointless complication. What you wind up seeing is it puts a lot more potential for information into the whole business here. Okay, so here we have our archetypal synapse here, going from left to right as always, and we've got the one on the left here with its vesicles, those little water balloons filled with neurotransmitter. Along comes the action potential. As a result, the vesicle moves to the cell membrane, merges with it, dumps the neurotransmitter into there. As you might guess, that actual process of exocytosis, dumping the neurotransmitter, is vastly more complicated, but this is our general model. And what people began to realize was that all sorts of neurons, all sorts of neurotransmitter types, in fact came with 2 different colors of neurotransmitters in the vesicles.
I cannot see the color here.
Kinds. Notice this is different from, oh, in this axon terminal, you release this type of neurotransmitter, and in this one, you release that type. No, every single axon terminal would contain both of these types of neurotransmitters. And notice also, it would not be one vesicle was a blue neurotransmitter vesicle and one was a red. Each one of them had a mixture of the 2 types. What has been found is that occurs in lots of different types of neurons. I believe the record is that there are now some vesicles that contain 3 different types of neurotransmitters.
So, question to ask and question to research in the future down the line is if the original idea from Dale was that each vesicle as opposed to one neuron. So, I'm going to do a research, like search, chat research later on, and what I'm going to ask hopefully is if that was the idea or contention as opposed to him basically saying that no, the vesicles basically has one form of like neurotransmitter in it embedded.
We've got here are 2 different classes of messengers coming out. And what you should be able to immediately imagine is that's going to produce a lot more potential for information. 2 different types of messengers. One of the characteristics that tends to be the pattern is that when you get 2 neurotransmitters in the same neuron, the same axon terminal and the same vesicle, they tend to be structurally very different sorts of classes of neurotransmitters. One might be a type made from a simple single amino acid. Another one is somewhat of a complicated protein. They tend to have fairly different structures. And what that often tells you is they're going to have different mechanisms of action. They're going to have different speeds of action. What you wind up seeing is very often when 2 different types of neurotransmitters are contained in a neuron, one of them works much more rapidly than the other. One has a rapid short-term effect, quickly decrementing. The other will have a longer effect, and by now you could begin to imagine what counts as a longer effect. You change gene transcription in that neuron. You change structural stuff happening, as opposed to the short one where just suddenly some little ionic excitability change occurs in a couple of milliseconds. You begin to see this pattern of 2 different ways of coding for different types of information. What we'll see shortly is one of the truly bizarre things that pops up a lot with these cases of multiple neurotransmitters is one of them will have receptors for it on the neuron itself. And we will focus on that shortly. That actually winds up making a fair amount of sense. Okay, so Dale trashed in terms of the notion of any given neuron having only one neurotransmitter. In some types of neurons, you have instead pairs, but following the same sort of rules. And thanks to the typical structural difference between the pairs of neurotransmitters, you tend to get different sorts of functions. You see a similar principle over at the endocrine end of things. A world of more than one single messenger carrying the same message or the same source of information.
I'm going to make note of this so that I can research like one messenger. Carrying multiple messages MSG. Yes.
Multiple messengers. Where we see here is what by now should be familiar as well. Here we got your pituitary, and you got your anterior pituitary, which is always facing left. And the anterior, here we have the glandular cells releasing, as you saw the other day, ACTH. Lots of this lecture is going to be about the regulation of the adrenocortical system, the glucocorticoid system, because it's the best hormone on earth, and I'm willing to prove it. But what we have here is ACTH coming out, and you remember what you had the other day.
ACTH, something he spent 25 years, ACTH hormone, pituitary gland. So going to look for that. I am.
Is incorrectly named. You've got CRH. That's that business about the brain as an endocrine gland, the brain releasing hormones into this little portal circulatory system, and out comes its characteristic pituitary hormone. So what you saw the other day is CRH is the hormone, the hypothalamic hormone at the base of the brain here, that's released into this local circulatory system, which stimulates these cells to dump ACTH into the general circulation. And that's the pattern you've gotten already of a neuroendocrine axis. The brain releases a hormone, into the local circulation, which stimulates the pituitary to release atypical hormone in response to it, which then goes and does something or other to a distant gland. You've got that one already. You have follicle stimulating hormone releasing hormone up here, which triggers follicle stimulating hormone, which then does something or other to your follicles or to your sperm if you don't have follicles, and these sort of three-step cascades. What we see with the CRH system here.
I think the three-step cascade is a great term, apt term. It tells you exactly what is happening here. I only very vaguely, ambiguously remember something along these lines where the brain would send release chemical, and that chemical would reach one of the glands in the brain, and then the gland in particular would modulate that and make it a different form of endocrine neurotransmitter, basically, which would go into a different space and work. It's magic.
One example of how you can also have a version of multiple messengers. Turns out CRH is not the only hormone coming out of the base of the hypothalamus that could release ACTH during stress. And instead, you've got a whole array of these. There are neurons in the hypothalamus that instead release vasopressin into the circulation here. Others that release oxytocin, others that release norepinephrine, others that release epinephrine, and collectively what they're doing is stimulating the release of ACTH. You have an entire array of these.
Wait, so the brain releases oxytocin and other kind of drugs, and all of them collectively end up in this process. What? Did I understand that correct? Okay, let's read this first. The big picture. The stress axis you already know. This part you mostly have right. Hypothalamus, pituitary, adrenal, HPA axis. Okay, Okay, hypothalamus brain releases CRH, corticotropin releasing hormone. Okay, corticotropin releasing hormone. CRH goes into anterior pituitary. The pituitary responds by releasing ACTH, adrenocorticotropic hormone. Adreno Adrenocorticotropic hormone. ACTH goes into the bloodstream, adrenal cortex. Adrenals releases cortisol. Adrenals release cortisol. Stress affects all over the body. Key idea: CRH does not become ACTH. CRH is a signal. ACTH is a new hormone made and released by pituitary cells in the response. Think CRH presses a button. ACTH is the output. Number two. Now the crucial anatomy Sapolsky is hinting at. This is where your confusion is coming from. The pituitary is actually two different glands stuck together. Anterior pituitary, true endocrine gland, makes hormones, responds to releasing hormones from hypothalamus, uses blood vessels, not directly neurons. Okay. Posterior pituitary glands, neural extension of the brain, does not make hormones, just releases hormones made by the hypothalamus, uses direct neurons. This distinction is everything here. The triple truncate cascade, not crate. What he's describing is really three parallel control systems coming out of the hypothalamus. The endocrine trunk, hormones, blood, pituitary. Example: CRH goes into tiny portal blood vessels, hits the anterior pituitary, causes a release of ACTH. Neural trunk, direct wiring. Hypothalamus sends axons straight to posterior pituitary and neural trunk. Okay, so this is like the posterior pituitary aspect. Releases oxytocin and vasopressin. These hormones are not modified, they go straight into the bloodstream. Neurotransmitter trunk, brain to brain signaling. Dopamine, serotonin. Okay. Norepinephrine mostly act within the brain, sometimes modulate pituitary indirectly. So Sapolsky is not saying all of these do the same thing. He's saying that hypothalamus is a master dispatcher using different delivery systems. Now let's place the specific chemicals you mentioned. CRH made in hypothalamus goes into anterior pituitary, causes release of ACTH, stress specific. Oxytocin made in hypothalamus send down neurons to posterior pituitary, releases unchanged into blood. Function, bonding, childbirth, lactating, and social behavior does not make ACTH. Dopamine, special case, form in the hypothalamus anterior pituitary inhibits prolactin. It's a hormone and neurotransmitter. Does not make ACTH. What is a neurotransmitter and what is a hormone? Distinction between that is something that you want to learn. Mostly acts inside the brain, can influence hypothalamus, indirectly modulates hormones release, does not go into the pituitary to become ACTH. Your key question answered directly. Are oxytocin, dopamine, serotonin going into the pituitary and making ACTH? Absolutely not. Only CRH and a few closely related peptides tell pituitary cells to make ACTH, its own target cells. Each thalamic signal has its own target cell, its own pituitary hormone, and its own downstream organ. Hypothalamic signals: CRH, STH, TRH, TST, thyroid, GnRH, gonad, dopamine, prolactin, or oxytocin released directly uses breast. Okay, oxytocin. I thought oxytocin was what um, prolactin. I always thought was associated with milk for some reason. I have no idea why. And dopamine, I thought was the happy hormone. Apparently, it does other stuff like it is like prolactin. Okay, prolactin mammary glands, kind of associated with that region. And oxytocin releases directly after uterus and breast. Final targets. I thought these hormones were supposed to make us happy. Hey guys, once it is Sapolsky's summary, the hypothalamus is just a stress center. It's a router. It's not just a stress center, it's a router, choosing between hormones, neurons, and neurotransmitters to control the body. What I'm going to do is dictate again. Hey, could you tell me the distinction between hormones, neurons, and neurotransmitters? If next, okay, no, not.
Work in the 80s went into sorting this out. And what's going on here, what you have are stress signatures. Different types of stressors will trigger different orchestrations of these ACTH releasing neurons up there, different orchestrations as to which hormones bring it about. And it'll be things like low blood pressure, hypotension will tend to trigger CRH and vasopressin. Low blood sugar, I think, was CRH and epinephrine and norepinephrine. The whole point is that you get a signature up at the hypothalamic level regulating this release of ACTH. Why have all these different ways of doing it? Two advantages. One is, depending on which orchestration of these you use, the shape of the ACTH secretory curve will be different. It's the same deal here. You use two of these to get a short, rapid effect, a more prolonged one. Here, depending on which mixture you will have a different profile of ACTH secretion. So you get control on that level.
Do not understand neurotransmitters. I'm just going to read this and like, if possible, maybe I will ask the inbuilt video summary to summarize me like what is the trace signal that he's alluding to talking about. Neurotransmitters are chemical messages neurons use to talk to each other, talk to other cells across tiny gaps. Hormones are chemical messages released into the bloodstream to affect distant. Hormones the message, okay? Neurons the hardware, okay? Neurotransmitters whose chemical whispers, local chemical whispers. Examples: glutamate, GABA, inhibition, not inhibition. Dopamine, serotonin, norepinephrine, what defines neurotransmitter releases from a neuron, okay, acts across a synapse. Oh, it operates within the synapse. Look, so for if this is a like neuron, it works in this tiny synapse. Works in milliseconds, target cell in the right next door, same dopamine molecules in the brain, neurotransmitters in the blood, hormone. Okay. Dopamine is a neurotransmitter as well as a hormone, is the idea. Hormones are released into the bloodstream. Examples: cortisol, insulin, estrogen, thyroxine, ACTH. What defines the hormone? Secreted by endocrine cells. So they're like special cells for this kind of thing. It travels via blood, reaches many tissues, only affects cells with the right receptor. So you have to have a the apt idea or metaphor analogy is hormones are like radio broadcast, not phone calls. Okay. The twist: some chemics are both. This is where Sapolsky loves to mess with people. Okay, it's like a good humored emoji. Dopamine, neurotransmitter in brain, reward and movement. Hormones in the hypothalamus. Hormone in hypothalamus, pituitary blocks prolactin. Okay. Norepinephrine, a neurotransmitter tension and arousal as a hormone released from the adrenal medulla as a hormone. As a neurotransmitter, it is like dopamine reward, and dopamine detox. When we talk about stuff like that, the phrase dopamine detox is basically talking, it is a neurotransmitter at that point. Just like norepinephrine, neurotransmitter, attention and arousal. Oxytocin, neurotransmitter in the brain, bonding. Oxytocin helps you bond. Okay, hormone in the blood, labor, lactation. So the distinction is not the chemical, but route, speed, and target. Okay. Where the hypothalamus sits, the bridge is a special bridge is special because it can one, use neurons, release neurotransmitters; two, use blood, release hormones; three, use neurons, dump hormones directly into the blood, posterior pituitary. One clean comparison table. Okay, what is it? Neuron, SL, that's by axiom. Okay, neurotrophic chemical, a chemical, synapse, blood, fast, slow, high, low, specificity. Examples in order to okay, I understand it.
Other thing is that each one of these not only helps to release ACT.
Okay, what I'm going to do is read from trigger ACH with low blood sugar. I think at the CRH, the epinephrine neuropinephrine, the whole body is that you get a signature up in the I'm going to pause now. Pause and then probably just going to ask ChatGPT or Gemini what he is talking about in this portion and then return. There's like mic going off in the background. Okay, welcome back. And we are trying to make sense of the portion from I think minute 13 to minute 14 when he talks about different traces and signatures. I did not understand, and I definitely do not understand like these two graphs, like this one and this one. I do understand this is like anterior pituitary, and these are different neurons. No, no, not definitely not neurons, either neurotransmitters. Yes, neurotransmitters, and neurotransmitters are coming here to cells perhaps, and this is where it is becoming full-fledged hormones and going elsewhere. And this graph, if you could see, was something along about like spiking up and down, etc., etc. So what he means by this is, first, different races slash signatures. When Sapolsky says things like you get a signature up at the hypothalamic level, or talks about different orchestrations and mixtures, he means this particular thing. Different stressors activate different patterns of signals in the hypothalamus. So different stressors, you could have different levels of stress. You could be like attacked by a real life threat. You could be having you could be hungry. You could be like you could cut your fingers, etc., etc., activate different patterns of signals in the hypothalamus. So in the hypothalamus, which is this region, this is the hypothalamus and this is the pituitary gland. Different responses are going to affect. It's not a one-off stress, as is written here, but which combination of signals, how fast, how long, and in what proportion. Think of it like this: stress is not a single button, it's a mixing board, and I think they're going to use stress this system as a music system, and hypothalamus probably being as a DJ. We'll read into it. Number two, the key idea you're missing, and this is the heart of it: wrong intuition, very common. Stress, CRTH, ACTH, cortisol. So this entire process, um, done through this entire like system. This is a system, by the way, hypothalamus and pituitary gland. It's not a single one-off process. This like other things going on. Sapolsky's point: stress, specific combination of CRH, vasopressin, epinephrine, norepinephrine, etc., specific ACTH pattern, specific downstream effect. ACTH is the same hormone, but how it is released can be very different. Why have multiple ways to release ACTH? Sapolsky explicitly gives two advantages. Let's translate both. Number one advantage is control the shape of ACTH release. The shape of the ACTH secretory curve will be different. It means how fast ACTH rises, how high it goes, how long it stays elevated. And these examples are really key. Sudden danger, sharp spike, and quick drop. Because, like, the danger, if it is sudden, by definition, is going to subside. Ongoing hunger, slow rise, and prolonged. Makes sense too. Chronic stress, flattened but sustained, like it just goes on. Using CRH alone, one shape, CRH plus vasopressin, not prosin, vasopressin, stronger, longer. CRH plus catecholamines, fast and sharp. Some hormones ACTH, different time profiles. Same hormone ACTH, different time profiles. That's what he means by using two to get a short rapid effect, a more prolonged one. Okay. Advantage two, each signal does other jobs too. This part is subtle but very important. He says each one of these not only helps release ACTH, it does other stuff down there as well. Meaning CRH, anxiety and arousal and attention, vasopressin, blood pressure, and social behavior. So each release, for instance, vasopressin, I think we read earlier here, was associated to like hunger and stuff, but here we can see it does other stuff as well. Epinephrine, norepinephrine, heart rate, glucose mobilization, etc. So the brain can say I want ACTH and increase vigilance, or I want ACTH and glucose release, and I want ACTH and blood pressure support. Why would brain say any of this? I don't know. And it does so by choosing what combination of releasers to use. This is what he means by coding the stressor. When he says you get a way of coding what sort of stressors you want, he does not mean conscious coding. He means biological coding. Different stressors equal different hypothalamic fingerprints. Examples: hypoglycemia, this; dehydration, this; vasopressin is here; fear, norepinephrine; infection, cytokines. This pattern itself carries information. That's the trace or signature. Carries information. So do we get these in a chart, perhaps? Why is this evolutionary brilliant? Every stressor produces the exact same hormonal response. That would be disastrous. Imagine if, okay, big. You don't want one blood pressure spikes for exams, full immune suppression for hunger, same response to infection as well as fear. Instead, evolution built a system where ACTH is common endpoint, but the road you take matters. One clear metaphor: ACTH as a song and hypothalamus is a DJ. Same song, ACTH, different mixes, club remix, extended version, and acoustic version. Final note: the hypothalamus does release ACTH in doesn't. Okay, let me read you this. Retry. The hypothalamus doesn't release ACTH in just one simple way. Different stressors activate different combinations of ACH, vasopressin, and cytocholamines, and these combinations determine how fast, how strong, and how long ACTH is released. This allows fine control over stress response. At the same time, each of these signals has additional effects elsewhere in the body. So the brain can tailor the stress response not just in intensity but in function, depending on what kind of stressor it is. And I asked subsequently, like, give me some examples, please. So, example one, I'm not going to read through all of this. Sudden physical danger, almost getting hit by a car, hypothalamic mix is CRH, norepinephrine, and epinephrine. Little vasopressin, ACH patterns, spark, very spikes, and this is like two things. First is the mix itself, and then the spike factor. So very fast spike makes sense. Short duration, sharp on and off. Why this mix? Glutamate can act immediately, says induced cortisol follows shortly after. Act immediately was catecholamines there. Why is it written here, by the way? I have no idea. Extra effects and also does other stuff. Heart rate goes up. Okay. So this makes sense. So when we talk about, like, we're in danger and something is like stressing us immediately right in our face, what happens is heart rate goes up, blood pressure goes up, glucose release, and alertness goes up. All of these things happen whenever we are stressed, or like we are in the moment and something wrong is happening. We are very much stressed and like afraid, scared, etc., in danger, immediate threat is the keyword. uh, these physiological events take place immediately at that point. And we are here through this understanding how they come into be, how they work physiologically, etc. And the first example was interesting enough to prompt me to read. Second one, low blood pressure or low blood sugar. Fasting diabetes, skip meals. Hypothalamic mix, CRH, yes. Epinephrine, yes. Some norepinephrine, minimal vasopressin. Why, I wonder. STH pattern, moderate rise, sustained. Okay. Extra effect, epinephrine mobilizes glucose. Because this is what happens when you are starving, like you have to get glucose. Cortisol promotes glucogenesis. Okay. Suppresses insulin action. Why? What is insulin action? I do not know. One day I will, hopefully. Interpretation, energy crisis, keep glucose alive. Dehydration or blood loss, vomiting, diarrhea, hemorrhage. Hypothalamic mix, CRH, vasopressin, some catecholamines. ACTH pattern, strong, prolonged, reinforced response. Extra effects, vasopressin retains water in kidney, constricts blood vessel. Cortisol response, cortisol supports vascular tone. Interpretation, maintaining blood pressure and volume. Examples: public speaking and humiliation, hypothalamic CRH, norepinephrine, minimal epinephrine, little vasopressin. ACRH pattern, moderate, longer, extra effect, increases anxiety and vigilance. Norepinephrine heightens attention. Cortisol alters memory and emotion. Be alert, interpretation, and focused. Okay, I'm not going to read through this one. Infection, fever, fatigue, fever and fatigue. So fever and fatigue are part of that when we are attacked by some outside um,
The pituitary,
agent.
it does other stuff down here as well, so that you get a way of coding, okay, under this sort of stressor, you want to release ACTH and do something or other in addition. The way you bring it about is getting these two. With some other stressor, you want to secrete ACTH and bring about some additional step there, and you do it with these three, that sort of thing. So you've got control over the shape of the stress response and to fine tune it there. So here we've got this completely more complicated picture of all these different releasers of ACTH. In addition, just to make things even more complicated, there are clearly hormones coming in there which, instead of releasing ACTH from the pituitary, they inhibit its release. And they release their hormone into the circulation also. And people have had evidence for these corticotropin inhibiting factors for decades and decades and decades, and people have had an amazingly tough time trying to figure out what these things actually are. The best implicated molecule so far is a peptide called delta sleep-inducing factor. Think about that. You go to sleep, and that's a very good time for turning off your stress response. So That making wonderful sense there. The main point for our purposes is you've got bidirectional control. You've got a bunch of hormones that stimulate the release of ACTH. You've got at least one fairly well implicated that inhibits the release. There's no way that there's not a whole bunch of other ones doing the same sort of signature coding at the inhibitory end. And you got different ways of translating different stressors into different endocrine profiles. And of course, what that winds up meaning is you get stressed like your big toe is telling you you just burned it, and you are going to have projections to a different array of these than if you were getting stressed, you were thinking about mortality. That's going to be having a different projection profile onto these neurons. What that suggests is a huge amount of coding information going on in the brain and the spinal cord, producing these different arrangements of how you dump out ACTH and the other hormones. Okay, so that as a first complication. In these various neuro and endocrine systems, you can actually have multiple neurotransmitters coming out of the same neuron. You can have an array of different hormones that bring about the same general response, but they differ in terms of fine-tuning the system. Next elaboration, one now having to do with some spatial characteristics of how these systems work. And here we have the same exact neuron again, and now we deal with Dale's law number 1, which is, you know by now, you get the depolarization here, sufficient to get to the axon hillock, you get your action potential, all hell breaks loose, very exciting ionic events of excitation, all or none regenerating, go shooting down the axon to the axon terminals. Dale's law number 1 was you get an action potential started here, and it is going to result in the release of neurotransmitter from every single axon terminal. So Dale's 2 laws, an action potential causes neurotransmitter to be released from every single axon terminal of a neuron. Dale's law number 2 just trashed, and it's going to be the same neurotransmitter released from every single axon terminal. And Dale's law number 1 is held up reasonably well, except for some work by a guy, and this is one of these sort of mad geniuses of neuroscience, a guy at MIT named Jerry Letvin, who what he did was, as far as I can tell, he just sat around in a dark, sort of abandoned warehouse for decades on end, and about once every decade he would write a paper that would transform neuroscience. And he actually was like this. I met the guy once, and it was one of the more terrifying experiences I had. I was working in a lab where, like, I had to go get an oscilloscope from someplace and pick up one because ours was broken, and everybody agreed the place to find it was in Jerry Letvin's warehouse that he lived in because he lived with oscilloscopes. And going in there, and it was like basically pitch dark, and there was this like sweaty Sydney Green Street kind of like, you know, guy sitting there, and he was like in a ripped t-shirt, and he had been in there for decades, and he was like chain smoking and sweating in there because it was like 150 degrees. So he spent a large part of the last part of the 20th century inside that warehouse writing one paper per decade that was transforming. So this was his particular paper, and I think this one was from the 70s. And what he showed was that under some circumstances, with him lined up with his hundreds of oscilloscopes proving this, you in fact had a violation of Dale's law number 1, which was you can get blockades here or there that would stop the action potential from propagating down some of the branches of this whole axonal tree. In other words, a whole different domain of controlling the flow of information, more subtlety there. Neurons could regulate which of their branches actually were sending on the message. And remarkably little has been learned in the years since then as to how this works, let alone how common a phenomenon it is. Most people wound up ignoring it. What has since been shown also is the wave of excitation that could come in through different dendritic spines back at the end, that there are ways in which branch points can be sort of blocked on one side so that the flow is shunted in one direction on the branches and not the other. This is this whole unexplored world suggesting there's all sorts of regulation going on at these branch points. It's not simply the case action potential, and you were going to dump neurotransmitter from every single axon terminal.
Do not understand what Dale's law is, so what I have to do is dig for it. So this is like the simple chat GPT. Why this rule exists. Logic, not trivia. Neurons make neurotransmitters using specific enzymes, packages them into vesicles, transports these vesicles down the axon. It would be metabolically messy to make dopamine in one branch, make glutamine in another. So evolution went with chemical consistency. But here are the critical exceptions. This is where neurons lives. Co-transmitters, the big one. Many neurons release more than one chemical at the same synapse. Common pattern: fast neurotransmitter, neuropeptide. Released at the same set is usually released at all synapses, but release depends on finding pattern. Low frequency small transmitter only, high frequency mandatory versus fast modulatory. One chemical that acts fast, another that acts slow. Okay, rare development of literature, early development injury disease. What is not true? Important, does not release dopamine at one synapse. Give another clean summary. Statement: a neuron releases the same neurotransmitter everywhere. Mostly true. A neuron can release multiple transmitter, yes. Different synapses get different transmitters, usually no. Released can depend on firing rate, yes. This principle has exceptions, yes. Neurons are chemically loyal but chemically simple. Oh, okay. But not chemically simple. If you want next, we can clearly SSRIs work slowly, explain volume transmission, or link this to stress peptides, matter what. What should I do? I have two options. Number one is copy and pasting the portion I did not understand, or just like go with it. I don't have to get that much deep into Dale's law as of now.
Down goes Dale on that one as well.
I'm going to have to take him
Equivalent.
at the
Equivalent
face
over at the
value.
endocrine end. And here we have our pituitary, which now no longer has a brain connected to it. But you've got the theme by now, which is you got the hypothalamic hormone, and out comes the pituitary hormone. And there's a whole bunch of different pituitary hormones producing the acronym flatbread, peg leg, flat peg, go to your deathbed remembering that acronym because it will make you happy and fulfilled. But okay, so there's a bunch of pituitary hormones and whatever facocked acronym these guys came up with. But what we have here, just to make life simpler, is we'll focus in on four of them. Flat peg, okay, so we're missing some of them there. But we've got growth hormone that comes out, prolactin, ACTH, follicle stimulating hormone. I'm picking these completely randomly. Okay, in the simplest possible of worlds, you would have all sorts of secretory cells, glandular cells sitting in here, and each one of them is capable of secreting all of those flatbread hormones coming out of there. It could be doing that. And it would simply be choosing which as a result of which hypothalamic hormone is coming down the pike there. That's not what you see. Instead, you have specialized cells within the pituitary. There's one type that only secretes growth hormone, which causes somatic growth, somatic body, somatic something or other, and those are called somatotrophs. And don't memorize that. Ones that only secrete prolactin, which cause lactation. These are lactotrophs, corticotrophs, gonadotrophs, something or other. So all of these, in fact, have specialized cells. Within the pituitary, there are cells that specialize in releasing only one of these types of hormones. Okay, so you got into that. So what would be the next simplest thing going on? So here's the growth hormone releasing end of the pituitary, and there's going to be the ACTH releasing end. No, that's not what you see. Instead, there's a mosaic all across the pituitary of all the different flatbread cell secreting types there. All throughout it. Okay, so that's just this mosaic. Good. Somebody was sloppy at the sort of fetal end, gluing it all together and just scattered them all over. And what winds up happening is you get local little neighborhood effects. For example, here we have somatotrophs that secrete growth hormone, and one is in a FSH neighborhood and the other is in a prolactin neighborhood. And what you see is you throw in the hypothalamic hormone, which causes growth hormone to get secreted, and this particular cell is going to be secreting totally different amounts of growth hormone than this one will, because it's in a different sort of neighborhood. And what's the implication of that? There's
Implications,
all
yes.
sorts
So,
of communication.
the word of the day, or at least word of these portion, is the implication. Like, what is the implication behind different things that you're saying, spouting out, that I do not understand? For instance, the Dale's law, like what? Of the first law and the second law, what is the implication? Like, what is the challenge, and what is like Professor Sapolsky going on about? At least this portion I understand, like it's simple to follow. Like if they are in a particular neighborhood, they're going to excrete or produce more of that element, as opposed to if they are in a different neighborhood, they're not going to do as good as job.
On between the individual cells in the pituitary. And it depends on what sort of neighborhood each particular cell is living in, how it is responding to the hypothalamic signal coming down. Wildly complicating. What that lets you do instead is different areas of the hypothalamus will turf its hormones to different parts of the pituitary, where its particular cell targets will be living in different sorts of neighborhoods. Simply more regulation, more complexity going on there. Next theme that comes through in terms of elaboration. We all have the negative feedback concept by now that came through in a lot of throughout the lectures last week. The whole notion, you get excited, you release a neurotransmitter into your synapse. If you're a neuron, you got to do some regulation there. You have to clean up after yourself. You remove the neurotransmitter from the synapse. You break down the neurotransmitter. You have to finish the whole thing. You are the endocrine system. You are the brain, and you've gotten it into your head that you want your adrenals to secrete glucocorticoids, and you start that whole cascade in whichever way, and you need to know when to stop secreting those hormones up on top. You need negative feedback information. All of these biological systems are characterized by that. You have enzymes where this enzyme turns this into this, and how does it know when they should stop doing it when there's so much of this stuff building up that this inhibits its activity? You get negative feedback regulation. You're making a lot of
Enzymes so has dual effect. You need to produce something, enzymes will help you produce that, to the extent that you have produced so much of that thing that it is going to hinder the production of enzymes, and then you're going to stop.
and whatever is making it has to be able to measure the levels of X. That is the simple rule with all this negative feedback stuff.
Negative feedback
So a
stuff.
first example on the neurobiological level. So what we have are what are called autoreceptors. What I inferred before, which is this weirdo world in which you not only will have receptors for a neurotransmitter exactly where they should be on the postsynaptic neuron on the other side of the synapse, but you will have them on the neuron that's releasing the neurotransmitter. It is an autoreceptor. It is a receptor right there on it. What's it doing? It's for bookkeeping. What you have is some sort of rule. If the neurotransmitters come pouring out of there and most of them go floating across, let's assume these are only red neurotransmitters here and bind to their red receptors there and do their thing to the next neuron. And just thanks to the floating around random life in the Brownian synaptic sludge there, a certain
Shout
number
out
of
to
them are
Brownian
going
motion,
to instead bind to this one. And all there has to be is some sort of rule in this presynaptic neuron that for every time one of these hits here, it means I've released 1,000 copies of this neurotransmitter, 1,000 molecules of it, and that's how I keep track of the numbers. And I will have a rule that if it gets below a certain level of my picking up this bookkeeping signal, that will be a signal to start making more of the stuff. If I'm getting too much of a signal, decrease the release, feedback regulation along those realms. This is where you see one of the elaborations on this often see is one of the neurotransmitters will exclusively work on a presynaptic autoreceptor.
It's just
That
too
one
big.
is
Could I have not like shortened this to this extent? Yeah, looks good. Probably. Like, do I have to show that much of my face for this videos? I don't have to, I guess. Let's go.
There merely in order to do the bookkeeping. You would think neurons might have figured out a more direct way of keeping track, like how many vesicles they dump, but instead this theme is there's some presynaptic autoreceptor which tells you on some statistical basis every time we get buzzed at this end, it means we've buzzed these guys 100 zillion times, and that's how we keep
This
track of
tally
how much
and
we want to make.
track
Negative
keeping
feedback loops
stuff.
there. You then
Like,
see the exact same equivalent in endocrine systems, which is all this negative feedback stuff. Something that was underemphasized in the lectures, the endocrine lectures last week, has been this element of neuroendocrinology. What you mostly heard about is what's going to be most dominating in the classes to come, which is hormones get into the brain and change how you think and feel and behave and all of that stuff. But also some of the time what hormones are doing in the brain is letting the brain know how much hormones there were in the circulation in order to do the bookkeeping, the negative feedback regulation. Your brain decides it wants to have this much growth hormone in the bloodstream, and thus it releases its hypothalamic hormone, which goes and all over the pituitary, gets those somatotropes to release growth hormone, and it does this thing elsewhere in the body. And the brain has to be measuring some consequence of that growth hormone doing its thing, measuring up there in order to figure out, have we gotten where we want to yet? Same thing with prolactin, same thing with every one of those. You have to have negative feedback regulation. So what do you need to do to pull that off? You need to have a part of the brain that is sensitive to that hormone signal. Sensitive in some sort of quantitative way where it can, in effect, count how much of the stuff there is in the bloodstream using the exact same sort of rule. If I've had one of those hormones in the circulation come and bind to one of my receptors, it means I've released 100 billion copies of it thanks to my starting this whole cascade. Did we want 100 billion? Did we want 107 billion? Did we want 93 billion? What do we do now? Does that tell us we've completed what we want? You get this negative feedback signal there. So the first thing you have to have is cells, parts of the brain that will measure, that will be responsive to a hormone signal. Those cells have to be able to have some kind of set point rule in there. This is the point of life that I am at, the point of my menstrual cycle. This is the point of reaching adolescence. This is the point of, am I stressed or not? This is the set point. This is the amount of hormone I would like to have in the bloodstream, this particular type of hormone. And what you need to then be able to do is, if levels have not reached that set point yet, you send a stimulatory signal to the hypothalamus, keep doing what you were doing, we need to push the levels up higher. And if the levels reach here or get even higher, you need to be able to turn this into an inhibitory signal going to the hypothalamus. So what you see are all sorts of regions of the brain that are sensitive to these various hormones, not just in terms of hormones affecting all the behaviors we're going to hear about, but also negative feedback regulation. So a way now of showing just how much more complicated it can be. So what would you assume is the general rule? Here's an example of, okay, let's make it simpler. Okay, so what would you expect to see? You're able to measure CRH coming out of the base of the hypothalamus, and the deal is something stressful occurs, and so it's pumping out CRH, and at some point you got as much glucocorticoids in the bloodstream as the brain would like for that sort of stressor, and that leads to the negative feedback signal, which stops the hypothalamus from releasing CRH. What sort of measuring is the brain doing? Most obvious version would be what the brain does is measure how much glucocorticoids are in the bloodstream. And a simple rule, the more glucocorticoids there are in the bloodstream, the more likely levels are to have reached the threshold, the set point that you want. So the higher the levels, the more of a negative feedback signal, and thus the less CRH being secreted. Totally straightforward, logical, measuring the level of hormone in the bloodstream. And this is how most endocrine negative feedback works. Measuring how much of the stuff, the more stuff there is, the greater the likelihood that you shut down the system, you put in the negative feedback signal. But in addition, there's a whole other domain of glucocorticoid negative feedback regulation. Where in this domain, what the brain is doing is not measuring how much glucocorticoids there are in the bloodstream, but measuring the rate of change, the rate at which levels are increasing. And that's a totally different domain of information there. Now what you're doing is measuring how many units of increase per second are you getting. And what you've got is this bizarre world there where you go from 10 units of glucocorticoids in the bloodstream to 12 units in 1 minute. And that means the same exact thing as going from 1,010,000 units to 1,012,000 units in the same length of time. It's not measuring absolute levels, it's measuring rate of change. And the faster the rate of change, the less likely CRH is to get secreted. And it turns out there's some domains of stress responses where what the brain is paying attention to is rate of change of hormone in the bloodstream. There's other circumstances where it's paying attention to absolute level. This tends to be what the brain listens to very early on in a stress response. This tends to be the more delayed response, but totally different dynamics there. Cells that measure the amount of hormone, that's not that hard to imagine, number of receptors, stuff like that. Cells that measure the rate of change, where going from 10 to 12 is the exact same thing as going from 1,010,000 to 1,012,000. What is that wiring going to be like? To this day, nobody has a clue. This was first sort of figured out in the 1960s. Amazing scientist up at UCSF named Mary Dahlman, who just sat and out of sheer just modeling work in terms of endocrine systems, predicted with glucocorticoids there are going to be two different domains of feedback. There is going to be a rapid rate of change sensitive system. There's going to be a delayed level of hormone in the bloodstream system. And here's exactly why I'm predicting this. And she turned out to be absolutely right. And decades later, people still don't fully understand how a cell measures the rate of change of something independent of the absolute levels.
I understand the lack of absolute terms like rate of change. It came up many times in when I was studying finance, for instance. One thing could go from like the distinction between going from 10 to 12 and from 10 million to 12 million. It's like just like big difference. Okay, the rate is going to be probably the same because one has a baseline of 10, the other one has a baseline of 1 million. Okay, so that is the distinction, and I'm kind of just familiar with it. I'm not sure other people are, but it is like interesting, tricky and interesting. Also doesn't have any unit. Rate of changes do not have units because it's just a rate of change from one point to the other point. What matters is like which point are you starting from, and what would be the disparity between going from a smaller terrain or smaller plane to a higher terrain or a higher plane. Going from one to the next level, point A being a smaller terrain, going to the next point versus point B, which is like a like significantly higher plane, going from point B in that terrain.
The thing that makes life a little bit easier is it's different parts of the nervous system that do each type here. The very rapid rate of change stuff, in fact, is not even occurring at the brain. It's occurring at the level of the pituitary. And thus, it's not so much regulating CRH release, but ACTH release. Okay, complicated. The main point of that
Main
is
point,
even
yes.
something as logical as, ooh, how do you keep your toilet bowl sort of thing from overflowing the tank in the back there? You need a negative feedback signal. You need a way for the toilet bowl tank to measure how much water there is there and the flotation device, and for that to have a set point and a way of then transducing, reaching that set point into putting the lid on the top of the pipe that's generating water.
Transducing is a
Yeah,
nice
totally
word.
obvious logical negative feedback loop stuff. And then suddenly in these neuroendocrine systems, you get much, much more subtle, complicated things going on. In some cases, you have positive feedback. The more of a hormone in the bloodstream, the more you stimulate the system to do more. You get that at certain points in reproductive life histories where you have massive changes of estrogen, progesterone, and what you have will be transient periods where you have positive feedback. All of these somehow have to get translated into how cells are working. Very unclear. Okay, so negative feedback. Next elaboration, something going on at the receptor level. Something known as autoregulation. And you could probably begin to figure out what that one's about, and it will make perfect sense as follows. If somebody screams at you all the time, you stop listening to them. And what you have done is just down-regulated your sensitivity to this pain in the neck person. If they give a very large signal, you down-regulate
Shout out
your
to
sensitivity to the signal.
auto-regulation.
If a signal instead becomes very weak, very often.
Again, let me try this again. Shout out to auto regulation. I think where he's going with it, It's the idea of being something like loud is going on in the background and you're just attuned to it. It's like you're like in that. It is the idea of relativity. When like Newton travels in a train, he is moving at the speed of the train itself. He is not like the train is going fast. He's not like splattered all over the windshield from the inside. That is not happening because he's like relative with respect to the people outside, but he's moving at the speed of the train itself. He's moving inside of the train. He's experiencing, in other words, time differently.
You will increase the attention you pay to it. You are showing auto-regulation. You are, within the realm of neuroendocrine stuff, you will be changing the amount of receptor for a neurotransmitter or hormone as a function of the levels in the bloodstream. And thus you have this logic. If you get a huge increase in the levels of some hormone in the bloodstream, there will be a likelihood that various target tissues will begin to downregulate the number of receptors for that hormone or neurotransmitter. Conversely, levels go way down, they go right up, and this is a general feature of all these neural and endocrine systems. Why is that interesting? When this regulatory, auto-regulatory stuff screws up. In principle, it should work perfectly. Okay, for some reason there's a doubling of the hormone message coming through in the bloodstream under some circumstance where it's going on for a long time and that's not right, and somebody down at some gland down there is messing up and drooling out way too much hormone. So what are we going to do? We have no idea what's up with them, but you've doubled the level of hormone, those folks down there. Okay, let's have an auto-regulatory response. Let's cut the number of our receptors in half to compensate. You can see in a rough way that's going to compensate. So massively increase the signal thanks to some disease state, something weird going on, and do a compensatory decrease. Have some disease or abnormal state where levels of some hormone or neurotransmitter go way down. What do? Let's up the amount of receptor enough to compensate for it. That's great. Where problems occur is if you don't compensate quite enough, or if you overshoot, if you begin to get...
What his idea is, I need examples of this, okay? Okay, first, what he means by auto-regulatory response, auto-regulation, cells adjust. And I was looking, searching, scrolling for the crawling, okay, I'm going to only mess up and embarrass myself. Cells adjust the receptor numbers to keep signaling stable when chemicals level change. Think of receptors as volume knobs, the not light switches. If the signal gets louder, turn the volume down. If the signal gets quieter, turn the volume up. This happens with hormones, neurotransmitters, and growth factors. Example one: too much signal, receptor down regulation. Scenario: excess cortisol, symptom, chronic stress. Cortisol levels very high. Target cells say, "Whoa, too loud." Compensation cells reduce the number of cortisol receptors. Some hormone, fewer docking sites. Same hormone, fewer docking sites, basically. Result: cortisol effect feels less intense than expected. Partial normalization. Sapolsky's line. Let's cut the number of our receptors in half to compensate. Example 2: too little signal, receptor upregulation, dopamine deficiency, Parkinson's disease. Early stages: dopamine level low. Postsynaptic neurons say, "I can't hear you." Compensation: increase dopamine receptor number, become more sensitive. Result: normally signaling for a while. This is why Parkinson's symptoms appear later. Compensation makes loss. Compensation masks loss, masks and adjusts. Where things go wrong, suppose is the real point. He says there are problems occur. Where problems occur is if you don't compensate quite enough, or if you overshoot. This mismatch is the disease. Let's see how. Example 3: overshoot hypersensitivity. Scenario: chronic antidepressant use, SSRI. SSRIs increase serotonin levels. Brain compensates by reducing serotonin receptors. Then drug dose changes or stress shifts serotonin release. Now, too few receptors, normal serotonin feels insufficient. Result: emotional blunting, withdrawal symptoms, mood instability. This is not the drug being bad, its regulating lag. Example number 4, undercompensation, weak signaling. Scenario, hydrothyroidism, not hydro, hypothyroidism. Low thyroid hormone, body increases thyroid receptors, but not enough. Fatigue, weight gain, depression like symptoms. Some hormone, wrong receptors balance. Same hormones, wrong receptor balance. Example 5, I don't depression setup. What he's hinting at, this is the big one. Old idea, too simple. Depression equals low serotonin. Sapolsky's modern view, depression is the mismatch between neurotransmitter levels and receptor regulation. Okay, that is a big idea. Possible patterns, normal serotonin plus downregulated receptors, low serotonin, insufficient receptor um, upregulation, fluctuating serotonin, lagging receptors. The system is out of sync. Basically, this is the key phrase. The system is out of sync. It could be like you have too much or too little, or you have not the right amount at the right time. That's why SSRIs take weeks to work. Symptoms fluctuate. Two people with the same serotonin level feel different. Another beautiful example, drug tolerance scenario, chronic opioid use, massive opioid signaling. Brain down regulates opioid receptors. Result, need more drugs for the same effect. Tolerance, need more drug for the same effect. That is what is known as tolerance. Stop drug, too few receptors, withdraw. Signal response and outcomes, this is like a good summary of it. Final Sapolsky style takeaway. Disease often isn't too much. Okay, okay, I'm slurring. Why am I slurping my words? Getting my words wrong. Final Sapolsky style takeaway. Diseases, just disease, not the disease, not the set of diseases. Disease often isn't too much for disease often isn't too much or too little chemistry. Is the failure of composition or composition that overshoots. This is a deep idea he's setting up before we're talking about depression. If you want next, I don't want to go next.
Mismatch, and that's where you got problems. We will see, as you read in the Zebra's book, with hopefully great detail, the chapter on depression, that's
Hopefully, whose book?
a critical thing that's happening. Because we'll see with depression, what's probably wrong are levels of a few different neurotransmitters, serotonin, dopamine, norepinephrine, all of this is going to come soon. And all of the standard antidepressant drugs change the levels of these neurotransmitters in the bloodstream. And that's great, and we know just how it works, except there's always a problem making sense of that, which is you throw in some of these standard drugs, SSRIs, things of that sort. Don't worry about the details. All of this will come later. The main point being that these antidepressant drugs, when you throw them in, they're changing levels of neurotransmitters within minutes to hours. You get somebody who is deeply depressed and you start them off on an SSRI like Prozac, and they don't start feeling better for days to weeks. There's some sort of lag time going on there, and as people have sorted it out, what the common sort of conclusion is, these drugs are not working so much by changing the levels of these neurotransmitters. What they're really doing is, by changing the levels, they're eventually going to cause an autoregulatory change in the number of receptors. And these change within minutes to hours, these change within days to weeks. And that's what these drugs are probably doing. We will go through that. Just to give you a sense of how awful it is going to get, there's some reasons to think that some of these antidepressant drugs work not by changing the level so much of this neurotransmitter, or not so much secondarily causing an autoregulatory change here, but instead causing an autoregulatory change in these autoreceptors. Unbelievably complicated. We will come to that for our purposes right now. What that begins to tell you is that
We'll
certainly
get
the
into
amount
it
of
later.
hormone and the
We
amount of
have like 2 or
neurotransmitter
3 dedicated lectures on anti-depression, not just anti-depression. Depression itself has dedicated lectures on it. So is depression a dedicated series or lecture? I think there's like a video that I just I think was published like 4 years or 4 months ago by Sapolsky. He's older in that video, and it is on depression, so that video should cover this portion, like what the modern what the modern serotonin SSRIs, basically like drugs like Prozac, etc., the treatment is doing. Probably something harmful, perhaps. I'm going to put a huge question mark even at the formulation of the sentence that I formulated, so don't take my word at face value. I might be like not understanding his point.
The amount of receptor as well, and have big pathological changes in the levels of the messengers. The body attempts to regulate with autoregulatory changes, and a lot of what disease is about is overshooting or undershooting. So that's one realm within the nervous system, neurotransmitters pertinent to depression. A realm, what do we got here? Okay, within endocrine systems, an equivalent one is as follows, something that you see that goes wrong in diabetes, adult onset diabetes. One of the things you have is a number of ways things can go wrong, but what your body does is when there's glucose in the bloodstream, when you've just had a meal, when you have all these nutrients in the bloodstream, your pancreas can detect the levels of glucose in your bloodstream and secretes insulin. Sugar shows up in the bloodstream, glucose, you secrete insulin, and what insulin does is tell your fat cells to absorb the sugar and store it away. Great, but you've got a problem, which is that your fat cells are already full because you are a typical westernized human and you've been eating to excess, and what you have is
Sugar shows up in the bloodstream, glucose you secrete insulin, and what insulin does is tell your fat cells to absorb the sugar and store it away. Great, because you got a problem, which is like you're a westernized human. You've only been eating in excess, and what you have is fat cells that are full up, and they stop listening to insulin because they can't take it anymore, because they can't take up any more stuff and store it away. The cells begin to be insulin resistant, and we'll see in a minute how that works. So suddenly you have fat cells that are responding to insulin, and what happens to your pancreas says it is crazy. We're trying to get rid of sugar from the bloodstream, and it's not disappearing. The person you just ate Hershey bars, and their fat cells are already full, but we're not cleaning the sugar out of the bloodstream, so let's secrete even more insulin. And let's secrete even more insulin, and what happens to the fat cells? They say this is ridiculous. We're all full up, forget it. We're full up, forget it. We're not taking up any more of these nutrient things. And in fact, what we're going to do, we are going to decrease the number of our insulin receptors. We're going to down-regulate our number of insulin receptors, and pancreas freak out, and they secrete even more insulin, and you go and you down-regulate, regulate even more, and you get, and what you have is this downward spiral, because at some point your pancreas is working so hard to dump these boatloads of insulin into the bloodstream that your body is paying no attention to, that you burn out the cells in your pancreas that make insulin, and you now you've got yourself a real serious problem. What's the key of what's gone wrong here? Too much nutrients, and thus too much insulin, and the fat cells begin to say we're not going to listen to it, and they downregulate receptors, more insulin, more downregulate, and this is at the core of what goes wrong in diabetes. This is a great read. This is a like mind-bogglingly new idea and a new way to see how things work. So our pancreas makes insulin, which like helps us like here's the excess sugar. So uh, wait, what? Who makes where are the receptors? Are they on in the bloodstream? Okay, I'm just going to listen to him
fat
instead.
cells that are full up, and they stop listening to insulin because they can't take it anymore.
Fat cells. So fat cells have like receptors. Okay, are they not? Do they not?
They can't take up any more stuff and store it away. The cells begin to be insulin resistant.
Yeah, fat cells. He's talking about fat cells. There's like 41. Insulin resistance. So your fat cells, basically in your belly, perhaps, are becoming insulin resistant at that point.
And we'll see in a minute how that works. So suddenly you have fat cells that aren't responding to insulin, and what happens is your pancreas says, this is crazy. We're trying to get rid of the sugar from the bloodstream and it's not disappearing. The person just ate 8 Hershey bars and their fat cells are already full, but we're not clearing the sugar out of the bloodstream, so let's secrete even more insulin. and let's secrete even more insulin. And what happens at the fat cells? They say, this is ridiculous. We're full up. Forget it. We're not taking up any more of these nutrient things. And in fact, what we're going to do is we're going to decrease the number of our insulin receptors. We are going to downregulate our number of insulin receptors. And
Yeah.
the pancreas freaks out and secretes even more insulin as a result. And you downregulate even more. And what you have is this downward spiral. Because at some point, your pancreas is working so hard to dump these boatloads of insulin into the bloodstream that your body is paying no attention to, that you burn out the cells in your pancreas that make insulin, and now you got yourself a real serious problem. What's the key of what's going wrong here? Too much nutrients, and thus too much insulin, and the fat cells begin to say, we're not going to listen to it, and they downregulate receptors. More insulin, downregulate, downregulate, and this is at the core of what goes wrong in diabetes. So we see these autoregulatory changes, and they can emerge slowly. They can help explain why some of these drugs work. Almost certainly, they help explain what's wrong in some of these psychiatric diseases, metabolic diseases. What's the punchline there? Number one, the amount of a messenger is important, but the sensitivity to the messenger is at least as important. You have as much capacity to regulate this as this in these biological systems, and thus you have as much potential for screwing up. They help explain what's wrong in some of these psychiatric diseases, metabolic diseases. What's the punchline there? Number one, the amount of a messenger is important, but the sensitivity to the messenger is at least as important. You have as much capacity to regulate this as this in these biological systems, and thus you have as much potential for screwing up at this end as screwing up with the amount of messenger. Yeah, did I see a hand up someplace? Yes.
Good lecture. This has to be the payoff, the ultimate payoff of watching this lecture. That I got to learn how things work, especially the example of diabetes and how
Great. Okay.
diabetes and
Don't listen
how
to anything she just said, because she's just gotten to an incredibly subtle, complicated point about how this stuff works. Go and read and memorize and recite to your roommates the section on the zebra chapter about the section in zebras on depression, looking at the paradoxical things where at the end of the day, people are not sure. Say an SSRI, a selective serotonin reuptake inhibitor, what it does is it blocks the reuptake of serotonin, and thus you wind up with more serotonin in the bloodstream. Ooh, somebody feels less depressed. I bet their problem was they didn't have enough serotonin. Work through the logic of all this autoregulatory stuff going on, and people still are not positive. that the problem with
auto-regulatory. I
depression
think
is
the
too little
key
serotonin
word
or too
here
much.
is
Because depending
auto-regulatory.
on whether
Word
the
of the day.
auto-regulation partially compensates completely, whether it overshoots, and whether it's these receptors or these, will completely determine whether the problem is too much or too little of a neurotransmitter. Incredibly complicated, exactly some of the complexities there. And that is so complicated, in fact, that it is essential that everybody stand up now and go to the bathroom for 5 minutes. Okay, again, back from the other flatbread day, your pituitary is releasing all sorts of different types of hormones under the control of all sorts of different types of hormones signaling from the brain. What you get is the pituitary is not just made up of one type of cell that could secrete every single type of hormone in response to each type there of messenger from the brain. Instead, you got specialized cells. You've got types of pituitary cells that only secrete prolactin in response to what's happening there, types that only secrete ACTH, which secrete luteinizing hormone, follicle stimulating hormone, so on. Okay, so they're specialized cells. Rather than having one part of the pituitary, which is the we're in charge of secreting growth hormone neighborhood, and one part that's the we do prolactin, and rather than it being broken up like that, instead it's just a mosaic of all the different types are scattered throughout there. So that would initially just seem like sheer sloppiness, okay, embryology, they didn't quite get it together to have discrete neighborhoods, that sort of thing, but it's just scattered there. What's interesting about that is you will see any given cell in the pituitary will go about its business, which is responding to its specific hypothalamic signal by secreting whichever hormone it specializes in. Every single cell in the pituitary going about its business will be a little bit more or less sensitive to its hypothalamic signal, depending on what sort of neighborhood it's living in, depending on which other types of these cells it's surrounded by. So that you will get, for example,
We covered this point.
FSH secreting cells, when they tend to be in a GH neighborhood, are far more responsive to their signal than when they're in an ACTH neighborhood or one that's a hodgepodge of any of these. I don't know what the rules are, but what you've got there are increased ways for regulation of the amount of secretion by determining which way you, the hypothalamus, turf the signals. Are you aiming for these types? Are you aiming for these types? More regulation that way. How does that work? What it has to be is all sorts of communication going on amongst the various pituitary cells. So the pituitary is not just sitting there passively responding to whatever the hypothalamus is saying. Instead, there's all this local regulatory stuff happening. One additional thing that was asked. Okay, so in this case, what we go back to
Why does it
is
matter though?
the
I
business
still
of
do not understand about the different cells being situated, like different cells at pituitary. I do not understand the effect of localization. Like what is the, how is the pituitary or how is the output rather in this case regulating itself through that? I understand from the body perspective, like you're secreting some out of pituitary and it's going all over the body, and if you have excess, that creates a negative feedback loop which gets back into the brain, and the brain tells you to slow down. Hey guys, slow down, pituitary. I understand that version, but what happens after that? And what point did you just make? No idea.
The more glucocorticoids in the bloodstream, in this boring level sensitive domain of you're just measuring how much of the stuff there is in the bloodstream, you're measuring there. Some part of the brain is measuring the levels of glucocorticoids, and the part is actually known. It's measuring there, and it has its rule. Once glucocorticoid levels get this high, I will send an inhibitory signal to those CRH neurons, and as a result, they'll secrete
And
less
then he is
CRH,
describing the
and
brain
that turns
portion
off the
here.
stress response. The more glucocorticoids in the bloodstream, the more of a negative feedback signal. What the person brought up was the very astute observation that, wait, what about all those other types up there, oxytocin, vasopressin, epinephrine? What that means is there are sensors that are talking to each one of those types there. And the brain at any given point, depending on the stressor, is saying, well, we want this much CRH, and we want this much vasopressin, this much oxytocin, and we're going to determine it all by measuring the level and the shape of glucocorticoid levels in the bloodstream so that there's feedback going on not just onto the CRH neurons, but to all those other types of neurons as well. Incredibly messy and complicated. Okay, nonetheless, very elegant. Pushing on, next version of complications. Now what we've got is another feature of how receptors go about their business. This general principle doesn't apply all the time, but in general, more of a ligand for a receptor. A ligand, for folks not familiar with this, a neurotransmitter is a ligand for a neurotransmitter receptor. A hormone is a ligand for a hormone receptor. The ligand is whatever the receptor normally binds. So this general, not universal, but general rule of the higher the ligand levels, the more likely you are to trigger this compensatory downregulation, the lower the levels, all of that. Here's another level of regulation that can go on.
Another.
So you got these receptors. They are complicated because their job is to bind their ligand.
I should have ligand.
And to then do
Remember the name ligand, okay? Never forget his name. He was the true soldier.
Other in the cell as a result, open up some channel that allows a change in excitability in the neuron, that sort of thing. These receptors tend to be very complicated. And what you wind up seeing is in lots of realms of receptors for different types of hormones, neurotransmitters, etc., you actually have the receptor made up of a number of different proteins, of a complex of proteins. So that, for example, okay, so for example, okay, here we have a receptor that's binding a ligand that looks like that, just like your premolars. And what you have there is it's got to come up with a complementary shape and lock and key, blah, blah, all of that. And in this particular case, this receptor complex is made up of 3 different proteins that are needed to pull this off. 3 different proteins, 3 different genes. A receptor being coded for by multiple genes. When you get the more fancy, complicated receptors, they tend to be complexes of more than one protein forming this pattern. So you've got multiple genes. That's interesting, and of course we're often running with that. What that means then is you've got the potential for variation, different flavors of the different genes, more different ways that this could appear. There's 2 different variants on this gene, there's 17 on this one, and you just do the combinatorial stuff, and thus you've got a huge number of ways of generating different versions of that same receptor, which of course will work a little bit better, or a little slower, or a little whatever, variation like that. Next complexity that you get with receptors. You can have a receptor that's made up of 3 different proteins, and there's 4 different genes that make proteins that can help construct one of these receptors. So you can make a receptor out of proteins 1, 2, 3, or 1, 2, 4, or 1, 3, 4, or off you go with that. Or you can make a receptor out of 3 copies of number 1, or 2 copies of number 2, and 1 copy of number 4. There's these possibilities of all these different subunits being different. Variability that way. So another realm of regulation is when cells change the subunits on their receptors. When cells will cause, say, degradation of, okay, so here we've got, so here we have this 3-protein receptor complex, and there's 4 different proteins that potentially plug into it, and this version has 1 copy of A, 1 copy of B, and 1 copy of C. Something may happen which will cause the neuron to degrade the copies of B and replace it with D. Or replace it with another copy of A, replace all this combinatorial stuff, and thus change a little bit how well the receptor is doing its job. And you see these subunit changes all over the place. One domain in terms of the neurobiology, neurotransmitter stuff, aspects of glutamate receptors. You guys heard something about that the other day. Glutamate receptors essential for learning, very complicated receptors, insanely complicated. Part of what learning appears to be about is not only increasing the number of copies of a certain type of glutamate receptor, but changing the subunit composition and making for a more responsive, more excitable version of that receptor. There's other realms with a different type of receptor where, in fact, you've got an abnormal subunit that's not supposed to be there, and as a result, this now produces a cell that is prone towards insanely high levels of excitation. This is a congenital reason for causing epilepsy. This is one of the genetic forms of
epilepsy. So is it what hardware failure looks like? So receptors are ligands basically, No, the ligands would be something that like binds with the receptor, and receptors are basically coded through DNA. DNA basically has the blueprint for coding, and there's like something about I have no idea, and this is where like ChatGPT would help me out. Um, that two or three proteins has sometimes come together to form or create a receptor, and they sometimes basically alter their creation slightly. I have no idea about the time you mentioned 124, 124. Where is yes, this version. So you can say a receptor out of proteins 1234 or 1234 of your go, or you can make a receptor out of three copies of number one or two copies of number two or one copy of number four. And there's these possibilities of all these different subunits being different. Variability, the way another realm of regulation is when cells change the subunit on the receptors. Yes, this portion. So this portion is absolutely key for ChatGPT to explain to me what is happening here. I can also take help of a book, a textbook on the subject. I feel like would cover these questions, answer these questions, cover these contentions, and that is the way I would fully understand this. So we are, I think, making progress in terms of understanding how epilepsy or congenial epilepsy, as you mentioned, or something that I personally perceive as a hardware failure occur, because the receptors are basically targeting for cells that are going to be much more excited or excitatory, or is it like through the alteration of different collective compounds of protein creating that cell itself, like where is it going, what is going on, and where is it going wrong exactly. At least he begins there, so I'm going to go to 5.5 and see what he's talking about here.
where you've got the wrong subunit winding up in another neurotransmitter and its receptor. All we're seeing here is lots of room for more regulation here, mixing and matching, changing the subunit stuff. That's how you begin to do that critical principle. Remember, all the stuff from the basic functioning, the flow of information from the dendrites to the axon hillock, what's that all about? No single dendritic input is enough to trigger an action potential. Instead, you have to have enough of them, enough summation to reach the threshold that the axon hillock has to initiate the action potential, and the threshold could change over time. Translate that into this. What does it mean when an axon hillock's threshold is changed? Those critical first channels that open up, you've changed the subunit composition. So that is a theme
No
all
idea,
throughout
no
the
idea
world of
what that is.
receptorology.
So if I am going to do something for future reference, this is the one thing that I have to do, and I will, I think, re-emphasize this point in at the end of the lecture as well, end of the session as well, because like this requires me to really delve deep into the topic, which I am not currently interested in doing, but maybe I should be interested in doing. So let's do it. I'm going to pause and go to my research. Here we go. The big question Sapolsky is answering here. He's not mainly talking about different neurological diseases in general. He's talking about how cells regulate how excitable or responsive they are at a molecular level. Okay. Let me read that. Read that again. How cells regulate how excitable or responsive they are at a molecular level. In other words, how does the brain subtly tune how easily neurons respond? How easily neurons respond or fire without rewiring the whole circuit. What does it mean? What does it mean to? The answer is by changing receptor structure or just receptor numbers. This is a regulation story, not a disease story. Okay. So, from what I read in this portion, what I understand is um, how easily a neuron would respond would depend on how the brain basically no, scratch that. How does the brain subtly tune how easily neurons respond, learn, or fire without rewiring the whole learn or fire. Respond, learn, or fire. So neurons respond, learn, or fire. Three tasks, not just one. By changing receptor structure, not just receptor number, not just receptor number. So this is astonishing. The first time I read this, I read the entire thing wrong. First one, receptors are often made of multiple proteins, but he says more fancy. Complicated receptors tend to be complexes or more than one protein coded for by multiple genes. Translation: many receptors are not single molecules. They're like machines made out of multiple parts, and each part is coded by a different gene. Think Lego receptor, Lego structures, subunits, Lego pieces, genes, instruction manuals for pieces. Okay. So already one receptor doesn't equal one gene. One receptor equals multiple genes working together. Why that matters? Genetic variation explodes. What he says, there's two variant of these genes, 17 and one of 17 of this one combinatorial stuff. Translation: each gene has multiple versions, at least. Okay, each gene has multiple versions, and I think one of the failure on my end is to think like these are like living physical structures. These are like molecular stuff, even maybe not with that. It's like yeah, mostly molecular stuff at the end of the day. Then suddenly you get hundreds and thousands of possible receptor versions, all technically the same receptor but functioning slightly different, faster, slower, more excitable, less excitable. This creates natural biological diversity, not pathology. The next level, same receptor, different subunit combination. When he says this, and that, this, and that, translation, even with a single neuron, the same type of receptors can be assembled in different ways, different subunit combinations, different properties, like same keyboard, different key switches, different key typing feeling. Okay, dynamic regulation, swapping subunits over time. But he says the neuron degrades subunit P and replaces it with T. Translation: neurons can remove one subunit, replace it with another, change how the receptor behaves. This can happen due to experience, learning, stress, development. So receptors are not static objects, they are adjustable machines. Where have I seen a receptor? I think we have seen a receptor even in something as simple as like two neurons like being in this position, okay, in the neuron terminal, I guess, in the synapse. So posterior and presynaptic and postsynaptic, etc. So we've seen receptors in that drawing somewhere like receptor drawing. Where's the drawing? We have seen it basically. Learning examples: glutamate receptors. What it says, learning appears to be about changing the subunit component. So two things. One is glutamate receptors. Glutamate is something that goes hand in hand in learning. Apparently, good to know. Learning appears to be about changing the subunit composition. Changing the subunit composition. Learning is about that. Okay. Previously, my guess would be like, oh, it's just like we are creating some variation in how our brain. Uh, no. I would say like my answer would have been like, how do we learn? Is we are creating stronger, more bolder, and stronger neurons. Okay, the neuron connections are getting like this thick when we're learning something. Learning is not just more receptors, it is also better receptors. Okay, but apparently learning appears to be changing the subunit composition. Okay, specifically certain glutamate receptors subunits make neurons more excitable. Learning strengthens synapses by swapping in those subunits. This is synaptic plasticity at the molecular level. Why epilepsy appears here? Important clarification. What he says: abnormal subunit, insanely high excitation, congenital epilepsy. That is an example, not the main argument. Here's the logic. Some mechanisms that allow fine tuning can cause pathology if it goes wrong. If wrong subunits are inserted or regulation fails, then neuron becomes too excitable, fires too easily, seizures. So epilepsy illustrates the cost of having such a power flexible system, not a tangent, a warning label. The axiom axiom hillock connection. This is the conceptual climax. What he reminds you of, no single dendritic input is enough threshold at axon hillock. Key neuro principle, the neurons fire only when inputs summate past threshold. Threshold is not fixed. His punchline, changing threshold equals changing subunit composition. Okay, so I think I remember this portion from his like teaching assistant's lecture, lecture 10 on like neurotransmitters, perhaps the part one, yes, neuroscience, like this one, this one. I think he mentioned that there is like an axon potential, axon helix in axon helix, and the signal gets passed when like we have an influx of positive charges outside of the neuron, and whenever we have receptors, it lets some positive charge come in, and like because the inside is negative, something happens, and if the threshold is surpassed, then the signal goes out, something like that, and then like the excess positive charges probably get out of the thing. This is how neurons adjust excitability, sensitivity, responsiveness without rewiring the circuit. When the he then generalizes beyond neurotransmitters, but he says same exact deal with hormone receptors. Translation: hormone receptors also have subunits, same combinatorial regulation, same tuning of sensitivity. Steroid receptors exception, steroid hormone receptors, often single proteins, act inside the cell, gene regulation, less subunit mixing, so different regulatory logic. The single general point he is making is the following. The brain regulates function not just by how much signal there is, but by changing the molecular makeup of the receptors that receive the signal. Okay, this is like cool and key and awesome. Adaptation explains adaptation, individual difference, vulnerability to disease, learning. So I'm going to reread this again because this is like so cool to read. The brain regulates function not just by how much signal there is in the body, perhaps yes, how much signal there is, but also by changing the molecular makeup of the receptors that receive the signal. So you have the brain has receptors, it's like this shape. It also works for better receptors. Okay, it like it like okay, it brings it creates, it propels, erects basically a better set of receptor, I think is the point. Flexibility comes from modularity. Pathology comes when flexibility overshoots. That's the intersection. No, I don't think I need this.
Channels that open and close and stuff where cells can change which are the pieces that make it up and change the properties subtly. Similar theme that could come through with hormone receptors. Same deal, a whole bunch of them are multi-protein complexes, exact same story. You could see something a little different when you have steroid hormone receptors. And as we saw the other week, remember it's got two domains, one which binds the hormone, glucocorticoids, just to pick a random steroid or estrogen, progesterone, or whatever, and one that binds to the promoter element in the DNA. We've got our if-then clause. If and only if this steroid hormone shows up, then you go and you activate that gene, our conditional clause. But what turns out to be the case is all sorts of steroid receptors, steroid hormone receptors, have various other proteins that they bind there. Cofactors is the term given for it, and you get different arrays of cofactors in different cell types. And as a result, when you activate this, it will do different things in different cell types. Cell type specific coding, and of course, the exact.
This portion I also need help of ChatGPT, but not now, I guess. Yeah, I'm kind of bored. What I'm going to do is I'm going to relay this to Professor Fink, and hopefully he will answer me.
Theme here, under some circumstances, cells will
But
change
these are
which
like
cofactors
important,
they
crucial
have
things.
holding on to those receptors.
These are like
So
really
what we see
important.
here are these additional layers of potential regulation. More complexities. Next one, Now we've hurdled past the notion of only one type of neurotransmitter per neuron. We've hurdled past the notion of the same action potential will be manifest in every single axon terminal. Now we see all sorts of additional information built around changing the number of receptors, changing the composition of them, regulating in those ways. One additional complication here with these receptors, which is a lot of them can bind more than one thing, can bind more than one ligand. And we see an amazing example of this at the neurotransmitter end of the world. Okay, you guys heard about the neurotransmitter GABA. GABA is the main inhibitory neurotransmitter in the brain. It is the workhorse for doing that. The GABA receptor, no surprise, binds GABA. Its ligand is GABA. It is found on the dendritic spines of GABA-responsive neurons, and the neurons just upstream of it will release GABA in response to its action potential and just goes about its thing. So what does the GABA receptor do? It binds GABA. And when it binds GABA, something or other happens so that this neuron becomes less excitable. It's an inhibitory neurotransmitter. It turns out the GABA receptor is insanely complicated. It is instead a receptor complex of a whole bunch of different proteins, and what the GABA receptor does is it binds some other things as well. 3 different classes, all of which are very interesting. The first class of additional things that it binds are things called major tranquilizers. What does the GABA complex bind? And here we have there's our GABA receptor, and GABA fits right in there. And it turns out the GABA receptor also has another binding site here for major tranquilizers. What are major tranquilizers? Barbiturates. This is how barbiturates work in the brain. Barbiturates, there are not barbiturate receptors. They bind to the GABA receptor. There's a side complex on it. There is a minor binding site on the GABA receptor for barbiturates. Okay, so GABA, inhibitory neurotransmitter, when barbiturates are around, does that increase or decrease GABA signaling? Okay, who says increase? Who says decrease? Who says what? Okay, me too. I wasn't listening either. Okay. I'll tell you, lurking around in the back during those TA's lectures, you really do get a sense of what interesting non-academic things are going on people's computer screens. But I digress. Okay, so what barbiturates do is they make GABA more inhibitory enough that you keel over and now they can slice you open for surgery. This is how one of the main classes of surgical anesthetics work. Then there's another binding site on the GABA receptor complex, and what that does is it binds the minor tranquilizers. And what are those? We've heard about those a bunch of times already. Those are the benzodiazepines, the Valium and the Librium, and they work there as well. And we've already heard about how anxiety disorders in some rodent lines are related to different versions of the gene for the benzodiazepine receptor. We've just defined it, what that gene is. That gene codes for one of the subunits of this massive GABA complex. So there's another domain there that binds benzodiazepines, the minor tranquilizers. What do? They increase the force of GABA signaling as well. Do they do it as much as the major tranquilizers? Of course that's not the case, because you wouldn't call them minor tranquilizers then. These guys potentiate GABA signaling a moderate amount. These guys potentiate it enormously. So now we have these additional factors binding to these GABA receptors. A third class, and this is in lots of ways the most interesting one of all, which is an additional binding site there that responds to a hormone, or a derivative of a hormone. What is it responding to? Derivatives of progesterone. Progesterone has a binding site on the GABA receptor. Progesterone is a steroid hormone. It's normally doing its thing here. And now we have it instead having a minor slot there on the side there that responds to progesterone. What does progesterone do? It also potentiates the effects of GABA. What's that about? 2 implications. One is, in the 1950s, one of the most common surgical anesthetics used was a drug called althecin, and it was a derivative of progesterone. People in the 50s would be anesthetized with this supposedly reproductive steroid hormone because it has this effect on the then undiscovered GABA receptor. It potentiates inhibitory GABA signaling. That's weird. Okay, so progesterone clearly,
Oh wait,
this relationship
they were like using it without verifying or knowing why they were using it or why it was effective.
evolved so that people could do surgery on people who didn't want to get major tranquilizers, and instead they went the progesterone route. Where is this relevant? It seems to have something to do with some of the mood shifts over the course of the reproductive cycle. There are reasons and pretty good evidence in lots of domains where perimenstrual syndrome, premenstrual syndrome, PMS, which is more accurately called perimenstrual, both before and after, what that involves clearly in some women is a shortage of progesterone, having some of these minor tranquilizing effects by way of the GABA receptor complex. So this is incredibly complicated stuff going on here, as you can see. Final domain, final interesting elaboration, which is now bringing in a whole additional concept and deserves a board all of its own. Okay. So here we have circular neurons, and what we've got is, this is the neuron we're interested in, what's it up to, and this is neuron A, and just to force some cognitive flexibility, this is neuron B going in the wrong direction. So we've got neuron B releases an excitatory neurotransmitter and thus excites A. Okay, this is not earth-shattering, it's probably releasing, say, glutamate, for example. So remember GABA, that inhibitory neurotransmitter we just heard about. So GABA does inhibitory stuff. So maybe there's a GABA neuron here sending its projection, and this one is inhibitory. Notice what we've done by turning neurons into spheres. We've lost all the little dendritic spines. So translate that into back to what it actually looks at. This is just schematic. Okay, so we've got two inputs. We've got an excitatory input by way of glutamate. We have an inhibitory one with GABA. What does that imply? Dendritic spines in this neuron contain both receptors for glutamate and receptors for GABA. That would be straightforward. That would be simple if that's how it works. That's not what you see. Instead, GABA neurons never send a projection onto what would be neuron A. What they do is they send a projection onto neuron B.
Well,
What's that about?
well,
What's that about is what GABA
Based.
is doing is making this neuron less excitable. It's working as an inhibitory neurotransmitter. It's making this neuron less excitable. What's happening here?
It's targeting
This neuron
your,
has just started an action
family.
potential that's coming down the axon, and thanks to a heavy GABA signal coming out, it silences it. It never gets to the end and dumps the neurotransmitter. This is totally weird. This is completely different from all the wiring you've seen already. What's bizarre about it? Number one, this is a neuron forming synapses not on the dendrites of this neuron. It's forming a synapse on the axon of this. And thus we have what is termed an axo-axonic synapse. What does that also imply? That there have to be GABA receptors sitting there. Autoreceptors, remember those are for measuring the amount of release, bookkeeping stuff. But here, this is perfectly conventional neuronal responding to a neurotransmitter, but coming at the completely wrong end with this weird axo-axonic projection. But what's most important here is seeing this implication. So what is it that this neuron does to A? What does this GABA neuron do to the excitability of A? And the answer is it does nothing. It has no effect on the excitability of A. What this GABA neuron does is alter the ability of neuron B to do something to A. This neuron is no direct effect here. What it does is it modulates the activity of this neuron. And thus you have a whole new class of type of communication. Technically, rather than working as a neurotransmitter, it would be termed GABA typically serves a neuromodulatory role in the nervous system. And that's this wonderful conditional clause, yet again, yet another if-then clause. GABA decreases excitability if and only if this neuron is trying to send an excitatory signal. All that GABA does is snuff out an excitatory signal coming down here. So modulation in that regard. We've already seen another example of that writ small. Back to the GABA complex here. What do the major tranquilizers do there? What do barbiturates do? They do not make this neuron less excitable. What they do is, if and only if there's GABA coming in, it makes the GABA work even better. The tranquilizers do not inhibit the neuron. The tranquilizers modulate, potentiate the activity of GABA. The minor tranquilizer, same thing. The progesterone, the same thing. So this is a different level of this concept of neuromodulation, if-then clauses all over the place here. So if and only if GABA is doing its thing, any of these minor ligands will potentiate it. If and only if this neuron is trying to stimulate here, GABA will have an inhibitory effect on the whole system. So you see lots and lots of this modulatory stuff. Final example, seeing the same principle of modulation. Now
and I will, by the way, shut down the recording anytime now. Just waiting for it to hit the 2-hour mark. 4 minutes.
So now we're measuring the amount of ACTH coming out of the pituitary. And back to that deal that we got all those different ways in which the brain can cause the release of ACTH. And starting off, we saw CRH. So suppose you throw in some CRH on top of the pituitary there, inject it into the circulation, and this much ACTH comes out. Okay, that's good. Now instead of putting on CRH, you put in both CRH and vasopressin, a second one of those. And what do you see? That's perfectly logical. You've got two things driving ACTH secretion. Now you put on vasopressin just by itself, and what you get is this. You get no secretion.
This portion I
Does
also do not
vasopressin
understand.
stimulate the release of ACTH? No, it doesn't. What vasopressin does is it potentiates the activity of CRH. It modulates CRH's activity. Vasopressin does nothing by itself. If and only if there's CRH getting to those pituitary cells, then vasopressin will potentiate its actions. All of those other releasers, the vasopressin, the oxytocin, epinephrine, norepinephrine, none of them are direct releasers on their own. They all are modulating, potentiating CRH's effects. So what we got here is this enormous realm of complexity of all of these if-then clauses, various hormones that are not directly causing anything, neurotransmitters that are not directly causing anything, ligands for receptors that are not either. What they're all doing is if and only if something else is happening, something else is going on, there's an additional ligand, they do their thing. Lots and lots of these conditional clauses, more complexity there. Okay, so what does all of this get us here at the end? Obvious. Lots of individual variability, lots of ways in which these systems are changing in response to experience, all that subunit changing business. Something happens and you get rid of one subunit, replace it with another, and you change the excitability of the system. All sorts of over and over conditional if-then clauses, tremendous increase in the complexity. These themes will come through again and again in the second half of the course. Okay, so let's stop at this point. Are there any questions? Since this was a major download, check the extended notes. Are the extended notes posted yet? Not yet. They will be sometime or other. Okay, any questions? Okay, so go read those notes as soon as they're up.
For more,
What I'm going to do is just the last portion. I'm going to do a full-on research. I think about from around 50-minute mark, and the rest of it needs to be researched thoroughly. I think 1 hour mark and then on. Like I want to know the implication of all these things. I understand most of it, not all of it. So that would be interesting. I do not have any other additional things to add except for a few things. That one, I require in this video to do justice to this session would require me to do a thorough research on most of the things, and it is really helpful to just ask ChatGPT to basically summarize what is going on in most of these cases. So I think I'm going to do that one last time because I'm going out. So maybe Canva as we have seen. So I'm just going to ask it to summarize it for me, and then leave the video and maybe read this result at a later date. Explain me this. I'm just going to hit 2 hour mark and quit recording. Okay, the single general point he's making, a most important regulation in the brain and body do not directly cause effects, they modulate effects that are already happening. In other words, no on and off, but if something is happening, then imply slash shape it. This is called neuromodulation in the brain and hormonal modulation in the endocrine system. I'm going to just keep it to this, and maybe just for the sake of it, like this is part 2. I'm not going to read here, but I'm going to read later. This is the key points. This is clever biology step 1, and then we have explanation going on here, this portion 4, and this is 5.6 final payoff 7 to all the way. So I'm going to conclude the video now. Thanks for watching.